In chapter 27, we continue our discussion of animals, specifically looking at vertebrate organisms. Just like our other animal diversity chapters and our other organismal diversity chapters, we're going to go through each of the defining characteristics that are unique to each of our new groups of organisms as we move through the animal phylogenetic tree. We're working our way up to mammals, and we'll get there. We'll learn about some different types of mammals, and then we will be done with our diversity unit. In chapter 26, we left off with the phylum Chordata. This is the deuterostome group along with echinoderms. Chordates are going to give rise to our true vertebrates, although the chordate group includes some invertebrates as well. All of the chordates, vertebrates or invertebrates, have a few distinguishing characteristics. They all have a notochord and a dorsal hollow nerve cord. As we move into our vertebrates, we'll see that these are going to become the vertebral column and the spinal cord, along with the well-developed brain. Our vertebrates are going to have pharyngeal slits. For the most part, pharyngeal slits are going to be present only in an embryonic form. They're not going to have pharyngeal slits. Not all of them anyway will have pharyngeal slits as we move through. Some pharyngeal slits are going to become gills. Others are going to become regions in the head and the neck. And then all of our vertebrates, most of our vertebrates anyway, are going to have a postanal tail, and we'll see that characteristic maintained through the majority of our vertebrate organisms. So we're not going to cover the invertebrate chordates. We're just going to move right on to the vertebrates. In addition to those chordate characteristics, our vertebrates are going to have an additional set of characteristics. Most of these characteristics are going to be derived from the chordate characteristics. We're going to build upon those characteristics to make more complex structures. So in addition to the chordate characteristics, vertebrates are going to have a vertebral column that is going to develop from the same structures as the notochord. That vertebral column is going to provide support. It's going to provide protection for that spinal cord, and it is going to allow the back and neck to move and to turn. And then the spinal cord is going to develop from what was the dorsal hollow nerve cord. The spinal cord is going to be the main neural system that runs from the brain, including the brain, all the way down, and then connects to all of the veins. I'm sorry, not veins, all of the nerves in the body. From there, we're going to see well-developed cranium. This is going to be the head with the brain inside. And then our vertebrates are also going to have an endoskeleton, so an internal skeleton that is going to be made of cartilage or bone. And then lastly, we're going to see something called Hox genes. So Hox genes are genes that are important for the basically the blueprints for organism structure. So the Hox genes are going to encode all of the different physical development, so things like legs and arms and wings, all of those different structures are sort of turned on or off by this set of Hox genes. Hox genes are actually going to be covered in our tissue chapters, chapter 32 and chapter 33. So depending on the semester, we may or may not specifically cover Hox genes. It just kind of depends on time. But as Hox genes diversify, we get more complex body plans. For your exam for this unit, you will need to be able to label the different groups on the phylogenetic tree. You'll also need to be able to label all of these defining characteristics. So which characteristics define which of these groups. Table 21.7 has a great summary of all of the different vertebrate groups. You'll want to be familiar with the characteristics that define each of these groups. These should all be in that phylogeny, but they'll also be in the lecture slides as we go through them. That phylogeny should be available in your study guide. This table may not be provided in the study guide, but you can always go back to your textbook or the lecture slides and look at this. Once we get past the chordate group and move into the vertebrate chordates, the very first group that we're going to come to, the oldest divergent lineage, are called the cyclostomes. The cyclostomes are jawless fishes, so they don't have a hinged jaw like you or me or many of the other fishes that we know of. There are tons of these in the fossil record, but most of them have since gone extinct. There's a couple of surviving lineages, and they would be representative of these early jawless fishes. So these jawless fishes are going to lack those hinged jaws, or any jaw really. Many of them have sort of a circular mouth that they can use for sucking or pulling. They don't have fins, and many of them don't have vertebrae yet. However, because in our phylogeny we are in the vertebrates, we need to recognize that they should have vertebrae. And this is because this first group that we're looking at, the hagfish, have actually lost their vertebral column. This is because this allows for increased flexibility, so they can kind of twist up and curl up in their environments. But they do have a notochord, and they have a cartilaginous skull. So here we're starting to see the flexible cartilage endoskeleton. So inside support, but still relatively flexible. These particular organisms happen to be blind, but they have a really good sense of smell. These guys are really gross. They produce tons and tons of slime as sort of a defense mechanism. So on their outside, they're just covered with slime. One of the other groups that falls into, one of the other fish that falls into this group are lampreys. So you can see here, this guy is jawless. He has this circular mouth filled with those teeth. So basically they latch on and kind of, I don't know, scrape the flesh off of whatever they're eating. You can see one down here on the bottom has sort of latched on sucker-like to that fish, and they basically just kind of grind out a chunk of the organism they're eating. So these guys don't have a hinged jaw either. They don't have any true appendages. They do have a notochord, and they have a rudimentary vertebral column. So these guys are have diverged pretty early, along with those hagfishes in the cyclostome group. Their vertebral column is not nearly as developed as some of the other organisms that diverged later. Our next group, moving along this phylogeny, are called the Gnathostomes. So these are the jawed vertebrates, and these are going to be most of the fish that you are familiar with. They have hinged jaws, and they have developed from the pharyngeal gills. So remember that one of the characteristics of our chordates is pharyngeal slits. And we said that some organisms maintain these slits, others lose them, and we only see them during part of their development. So most of our organisms that we're going to look at in our fish groups, well, all of our fish groups, are going to have these pharyngeal slits or pharyngeal arches. So the slit, if you think about the gills on a fish, I usually imagine a shark for this. If you imagine the side of a shark, they have like slits or openings on the side. Inside are the gills. So the slits allow water to come in. So those slits allow water to enter. And the arches, excuse me, are supports in between the slits. So they basically provide sort of an anchor for any flesh that's there. So in the jawless fish, they have the slits and the arches. And then in the early jawed organisms, we have the loss of some of these front slits and arches, and instead they are replaced with these jaws. And then as we move farther to our modern jawed fishes, we get a more complex jaw. So those pharyngeal arches, instead of developing into arches and slits, they actually develop into portions of the jaws. So as evolution moved through, some of the arches were lost, some of them were modified and are now part of the jaw. This was really important because it allowed these vertebrates to become predators, much more efficient predators. Once we have jaws, we start to get more efficient teeth, we start to get more powerful bites, and we have better advantage for predators. Our example of the earliest diverging gnathostomes or jawed fishes are the Chondrichthyans. So you know these as sharks, skates, and rays. Skates are kind of similar to our rays. There's a picture here. You can see they're kind of shaped similar and have some similar characteristics. Stingrays and skates are essentially flat sharks. Instead of having small pectoral fins that come off kind of towards the bottom, they have really big pectoral fins that kind of take up the entire side of their body. You're probably relatively familiar with the structure of a shark. We've got a dorsal fin up on top. We've got pectoral fins on the side, small pelvic fins, and then tail fins on the back. Shark fins, tail fins are going to be vertical. So they're going to be up and down. Our aquatic mammals are going to have tail fins that are side to side. So that's kind of one of the differences between our big aquatic predators. So our very early gnathostomes are Chondrichthyans. Everybody after and including the Chondrichthyans are going to be considered gnathostomes. Everybody that we're going to look at from here on out is going to have a jaw, a hinged jaw. A couple of key features for our Chondrichthyans. One, they are denser than water. If you're denser than water, you sink. And so these guys have to basically manually continue to stay afloat. So they have to keep swimming to maintain their buoyancy, and they have to keep breathing because breathing in is going to help them remain buoyant. They have a skeleton made of flexible cartilage. So what this means is that their cartilage is not replaced by bones. As we move on to our bony fishes, we're going to see that the bones get really hard because of calcium. Cartilage is essentially bone that has not been calcified. So you don't get this sort of hard bone structure, which allows these organisms to be a bit more flexible. Sharks are the earliest fish to develop teeth. Their teeth are not set in the jaw though. The jaw can still open and close. They do have a hinged jaw, but unlike you and I, whose teeth are set into our jaw, anchored into our jaw, Chondrichthyans don't have teeth that are anchored. This allows them to be easily replaced. So they can lose their teeth, constantly replace them. Basically, their teeth are kind of on like a conveyor belt. When one is lost, they kind of push forward and grow a new layer, and they just continue to be pushed into position. This makes sure that they always have teeth, but it also means that if they have a damaged tooth, it can be easily replaced. So instead of being anchored into the jaw, they're basically held together by connective tissue to the cartilage. We have a couple other just quick characteristics of our chondrichthyans. They have a dual chambered heart, which is a single circulation mechanism, basically in one side, out the other. They have really great sense of smell. This happens in a lot of predators. They have really good vision as well. So the two of those combined together are a predator adaptation. They need these strong sense of smell and vision to help find their predators in dark, murky waters, especially that sense of smell, because some of these are performing their predatory duties in really, really dark waters, and so smell is going to be really important. They use something called a lateral line to help detect their environment. We're not going to go into that for this class, but it's just kind of a really cool pressure-based way to sense their environment. And then our chondrichthyans have a variety of egg-laying strategies. Usually fertilization is internal, but then depending on the organism, they have different strategies for egg laying. In most semesters, you probably don't need to know this. We may not even cover it in class, but on the off chance that I do cover it, and because I might use these words a little bit later, these are some of the egg strategies that chondrichthyans have. Some of them are oviparous, which means they lay their eggs in a pouch, and then their pouch is deposited into vegetation so that it doesn't float away. So there's an image here on the right. There's a pouch with this little tiny dogfish shark inside of it, and it develops in there, and then it hatches just like any other organism out of an egg. We also have ovoviviparous. This is where the egg is retained inside the female, but there's no placenta. And then we have viviparous, which means the egg develops inside a uterus with a placenta. This placenta is not the same origin of the placenta in our placental mammals. This is basically an example of convergent evolution. So their placenta is a little bit different than the placenta in placental mammals. But the egg is developed inside the uterus with this placental material that is nourishing the young, instead of being contained inside of an egg, like say a bird. All right, so from the cartilaginous fish, we move on to the fish with bony skeletons. This is the most numerous type of fish. Bony skeletons basically means that they are a hard skeleton made of calcium, or calcium deposited onto the cartilage. There are two living clades, the Actinopterygii, the ray fin fish, and the Sarcopterygii. I'm not going to use those names because I can hardly say them. We're going to use ray fin fish and lobe fin fish. You can see the difference in these two structures. I apologize, these images are opposite. On the top, we have a lobe fin fish. You can see the bone structure here. We've got a variety of bones, smaller bones, and then even smaller bones coming off of those. Whereas the ray finned fish, we've got a kind of single layer of similar size bones with one single bone coming off of it. So the difference here is in the bone structure of their fins and how the muscles attach to the fins. So a couple of features that distinguish these bony fishes from our cartilaginous fish or our chondrichthyans um, are that they have a skin that is covered, the bony fishes, in a thin epidermal layer that produces a little bit of mucus. They have a structure called an operculum that covers the gills. So if you imagine a goldfish in a fish tank and you see that goldfish kind of opening and closing its mouth and its gills opening and closing, you can't see it, but I'm doing that as a demo right now. Gills, the flap around the gills open and close. So that is called the operculum, and it basically allows the fish to sort of wash water across their gills because that water contains oxygen that the gills are going to pull in. Our bony fishes also have a swim bladder for buoyancy. So this is different than our chondrichthyans, which are denser than water and have to keep moving and keep breathing to remain buoyant. It's a special organ that basically controls inside air pressure so that they can float or sink within the water column. And then they have a variety of different reproductive strategies. We're not going to go into all of them here, but many of them are the females shed their eggs into the environment, and then the males basically spray sperm on top of them, and the eggs are fertilized in the environment, although there are a variety of different ways that this might happen. So our ray-finned fish are most of the fish that you know. This is the largest clade of bony fishes. Pretty much any fish that you can recall off the top of your head is going to fall into this. Eels, sea dragons, cod, salmon, etc. Their fins are supported by these thin, flexible rays that come off of a single layer of bone from their skeleton. They have muscles that are attached back in the body, attached to the body and then connecting to the bone. So those muscles are sort of controlled by the bulk of the muscles within the body. On this fish here, you can see those rays coming off of their fin. And then you can't see it very well, but right here is an operculum. That is the flap that covers the gills that open and close to allow water to wash across the gills. So basically what happens is that the fish can open its mouth and water flows in through the mouth, across the gills, and then out. And then as water moves across the gills, oxygen is taken up by the gill surface and diffuses into the organism. In contrast, we have the lobe-finned fishes. Most of these have gone extinct. There's only a couple of them. Coelacanths are one of the types. You can see here it says that they were thought to be extinct. There are only 2 known species of coelacanths. So these are relatively uncommon fish. The other type of lobe-finned fish is called the lungfish. Now these are important. There's only a few of them, but they are important because it is out of the lobe-finned fish that we are going to get our tetrapods. And so we need to know a little bit about their fin structure and how it differs from the ray-finned fish. So their fins are supported by extensions of their pectoral and pelvic areas. So we got the body of the fish here, and then their fins are moved by muscles that are in the fin themselves. Coming from the fin itself are these rays, just like in our other fish, but they come from a different bone structure. Remember in the ray fin fish, they had a bone structure where they had basically a spray of smaller bones and then little tiny bones that came out of those. For our lobe-finned fishes, we have this set of bones, and then coming off of this more complex set of bones are these tiny bones that produce the lobe of the fin. You can't see them very well on these guys, but we've got fins here and here. You might still see the rays like you do in the ray-finned fish, but the internal bone structure is going to be a little bit different. So their movement is controlled a little bit differently. And in the next video, we are going to talk about the significance of this bone structure.